Hello friends, welcome to lecture 2 of week 10. In this lecture we look at the single cell genomics, how we can do this, what is actually single cell genomics, how it differs from tissue or any other biological material that we have been using before, right? How we can do this, what methods, techniques can be used in terms of the sequencing, or basically how to reach the single cell status for the doing the further analysis, and how the single cell gene expression or the concept of single cell and the outcome of the analysis helps in the human health. So basically single cells are microscopic cells and you cannot see a single cell. Like in your skin you can see the tissue, but you cannot see the individual cells that make up that tissue. And if you see the skin also, there could be several layers of tissues, and each tissue will contain cells, and basically all those cells, it has been assumed that they are doing the same function, same expression, same job, but it may differ, and the differences may be very important in context of diseases such as cancer. So basically these single cell is a smallest entity, microscopic, cannot be seen by naked eye, and basically when they combine they form tissues. In bacteria you may have a single cell, or you can have a cluster of cells called biofilms, right? So basically generally these single cells are characteristics of unicellular organisms, but when they accumulate and they have formed the multicellular organisms also, like eukaryotes, right? And basically these single cells may be doing activities on their own which may not be common across all the cells in that tissue. For example, this is a tissue that we can see, right? So it is colored because there could be like heterogeneity in the cells. So now for each location, if you see like this blue color region, if you see, you can see that there are if you just expand this, you can see there are multiple cells which are overlapping, right? And they may be having a differential gene expression. Genome has to be same, but methylation patterns may be different, right? And similarly, like you can segregate a tissue into multiple cell types, like you can see here. Okay, so basically in this picture you can see that a cells taken from a tissue have been segregated into multiple cell types, right? Similarly, in case of bacteria, a bacteria is dividing. So one bacteria forms two forms four, and like that. So now in this we generally assume that in a pure culture or in a colony of originated from a single bacterial cell, all the cells would be having same DNA, same transcriptome, right? But actually in case of bacteria, even the genome can be different, because the individual variation may exist between the various bacteria due to the differential error rates by the polymerase and many other reasons. So in the case of unicellular bacterial genomes, it may be even more important, like in case of biofilms we have seen. So basically when we want to do single cell genomics or omics in broadly, if you see, because it may include genomics, transcriptomics, metabolomics, proteomics, right? So in that you have to first segregate the cells either by laser capture microdissection or FACS or microfluidics. Then when you read the single cell, you will isolate the DNA from that single cell, label it by the adapters, and then go for the library preparation, and followed by sequencing or microarray based analysis or any other analysis, right? So this can provide a picture of what activities were going on in a single cell, but we have to be sure that while we are isolating a single cell from a tissue, we are not inducing any further stress or so that the expression level of the genes change. Like it has to be in their natural state so that we can capture the cellular activities correctly. So in this, for example, there is a termite. Okay, you might have seen termites, right? So when you isolate the termite gut, so inside the termite gut there are protozoans, and inside the protozoa there are single cells like these bacteria. Okay, so basically these are endosymbionts which have changed themselves according to the environment and adapted to that environment and doing something for the now the termite. Okay, so this studying these single cells could be one of the ways in which we can appreciate the importance of single cell genomics and transcriptomics. Similarly, in human intestine, if you see, like so intestine contains more than like 1200 species of bacteria, and like millions of numbers, almost 2 kilograms of your bacteria sits inside your intestine, right? So, and if you see the total number of cells in the human body, the bacterial cells may outnumber those, right? So, in the intestine also, when they are forming a lining or biofilm, right, or doing a job in the like large intestine, so there may be different cells, and like you may would look like to study each cell individually. Another example could be lung, where you can see that there is a like heterogeneity in the cancer cells, right? Similar in skeletal muscle also, you can see there is a heterogeneity in the type of cells. So each cell may be important and crucial for its own functioning and contribution to the environment, or affected by the environment, that we can capture in single cell genomics. Okay, so basically in single cell omics, we target the genomic DNA or RNA, and then follow the next generation sequencing strategies to sequence it to obtain the genomic content or the information on the transcript expressed genes, and that may vary according to environment, condition, or any treatment state, or longitudinal variations. Many things can be possible which you can study, right? And it provides a higher resolution of cellular differences and better understanding of the individual cell function in context of the microenvironment. Okay, and in cancers, the heterogeneity in the cells is very crucial to understand, to know that which of the tissue part or component of the cells are actually cancerous, and the remaining could not be, right? And even in those, there could be several, like in the cancer cell, there could be several variations among, in the vicinity also. So basically, the single cell genomics involve first of all the isolation of a single cell. Then a single cell may not contain as you know that it contains DNA, RNA, right? RNA is the transcriptome, and so, but that may not be sufficient to DNA quantity to make library and go for sequencing. So one round of amplification is needed, but before that there has to be strategies to isolate that single cell quickly in its native state if possible, and then basically look for the and then extract all the contents in a space, so that there is no contamination, and then put the conditions for the amplification of by PCR, or first for the RNA conversion to cDNA and then amplification, so that you have sufficient amount of DNA for the further analysis. But some of the modern methods can also go for direct sequencing of those DNA fragments or RNA fragments, right? So Illumina, Ion Torrent, MGI, even PacBio or Nanopore can also be used for the sequencing. So the RNA which is expressed in a single cell can provide you insights on how that cell is behaving or expressing itself in the context of its microenvironment. In microbes, a population may arise from the same species, appear genetically clonal, but there could be several differential RNA expression or epigenetic modifications that may lead to cell variability, right? Further, in case of endosymbionts, like I also showed an example earlier, there could be that the bacteria you cannot culture, but you have to isolate this cell directly, right? And then, like for example, you cannot culture the endosymbiont which is existing inside the protist in the termite in the previous example. So the strategy could be to isolate, handpick the cells from there, then break open those cells, amplify the DNA, and then go for the sequencing to reconstruct the genome. Okay, so basically now you can see here, like these are the tissue that contains multiple type of cells. Then you have to isolate each and every cell, get the DNA out of that single cell, right? Barcode the DNA, and then followed by the computational analysis. So this is basically the overall strategy of sequencing the DNA or RNA from a single cell. Okay, so here I am just briefly showing the workflow for doing a single cell genome. So first of all, from the environment you will be getting the tissue. Okay, tissue could be any eukaryotic, prokaryotic. In like eukaryote, if you say human, there could be several different tissues possible throughout the body. So you can take one of those which is of your interest. Then you can use either laser captured microdissection or FACS to sort the cells, or microfluidics to separate the cells, okay, like you do in the case of the chromium instrument. So then once you have got the single cells, you will isolate the DNA, label the DNA to make the library, and then you will go for the sequencing. Similarly, in epigenomics also, single cell epigenome may be of importance, and that can also be done here. Okay, so it is now a widely used technique in the modern biology to study the physiology and disease, right? And it can uncover a lot of genetic mechanisms or variations in cells that may be very crucial to understand cancer and basically response to treatment also. And many tools have also been developed to address the assembly or quantify the expression levels of the transcriptome from a single cell, like spares and many things we will also discuss. As we earlier mentioned, like in case of prokaryotes also and microbiomes or biofilms, these single cells are also like equally important, which is actually called as single amplified genome, because from a single cell, the DNA that you capture, right, you have to amplify that to get sufficient DNA material to go for the library preparation. Okay, so these it is not always that you will get all the complete genetic material, and in amplification also there is a chance that you may lose some of the fractions, but still this can from a single cell. But if you have multiple single cells, then it is possible to actually reconstruct the entire genome. Okay, like this is the example of a biofilm, okay, which basically you can see that there are different type of cells in that, labeled by different types, and then you can separate those cells by FACS, okay, then followed by mRNA or genome sequencing, okay, then you can also have that cultured, and then basically you can go for the single cell based on some treatment response or any study that you are doing. You can go for the microscopy as well as the genome amplification followed by sequencing. Okay, so in this picture basically we are showing that how the single cell DNA methylation sequencing can be done. Okay, so this we have also discussed when we were dealing with the epigenetics part. So basically single cells are sorted, and then for each cell the DNA that you get, the bisulfite conversion can be done, so that the C's, normal C's become T's, and only the methylated C's remains, and then you can go for the library preparation and sequencing to base call all the C's. Now whichever you find are actually those which are the methylated ones, because the normal C's you have already converted them into the T's. Okay, so single cell genomic here can tell that how the epigenetic differences or modifications could be different in the different cell types, or even in a tissue also there could be cells which may have different levels of the epigenetic modifications. Okay, so ultimately you are basically having any tissue, okay, then segregate the cells and differentiate, okay, isolate the single cells, go for the RNA or DNA. Okay, in case of RNA you have to convert that into cDNA, because RNA is not stable, and basically cDNA or DNA is the input for the library preparation. Then go for the amplification by RT-PCR or normal PCR, and again look for the various transcripts that were present by sequencing and further analysis. So this can tell like what transcripts were there, that means which genes were expressing at that point of time, what were the relative levels of expression, and then you can use various statistical methods to quantify and also compare the RNA expression between different cells or under different treatments or conditions or even in a same microenvironment. Okay, so the first thing that you will be doing is to isolate the cell before going for this. So there could be many ways, like fluorescence activated cell sorting. FACS is a commonly and traditional method of sorting the cells. Other is by micromanipulator, which is a device attached with a microscope in which you can pick single cells by looking under the microscope, okay, without actually killing the cell, right? And micromanipulators are good options because that gives you like something that you can do in your own lab and easy to use also, and people have also successfully used micromanipulators for single cell genomics, right? Then laser capture microdissection LCM is also method in which you can get a single cell, which we will see in the next slide also, that how you can from a distinct like or a very complex tissue you can get the cells. Okay, so basically for the FACS you have to prepare dilution and then go for FACS and sort the cells, okay, differentially. Or in case of laser capture microdissection, like this piece you want to cut, so by laser dissection you can cut those pieces, and then all the pieces you can separate and then go for the single cell genomics. Or in case of microfluidics, basically you can segregate the cells, right, and go for the sequencing of DNA or RNA. Even like treatment and other kind of studies can also be done. And micromanipulator, where you have under the microscope you can look at the cell, and by micromanipulator you can pick the individual cells based on the differential morphology, or if you have too many cells to deal with at that point of time, it may be difficult because many cells may have similar morphology and you cannot make out. But it is very good when you are not dealing with that much complex diversity of the cells. Okay, so microfluidics, FACS, micromanipulators, all are good methods for isolating the single cells and detaching from their environment, but that may cause some perturbations in the transcription profile in the RNA expression analysis, so which should be minimized, or the methods that do not cause these differences should be used. Okay, so now looking at like how many cells may be needed to do the single cell RNA sequencing project. So basically at least 10 million reads should be are needed, right, of basically you can say that 50 reads per kb per million should be good enough, so that you can tell that any transcript is being expressed. Okay, and now to get sufficient coverage of each and every region, for example a 1 kb long region, at least 500 reads should represent that region with a minimum coefficient variation of 4%, and typically like for a mammalian cell containing about 2 lakh mRNA, at least like sequencing data from at least 50 single cells need to be pooled in order to achieve this minimum C value. So a single cell may not be sufficient, and at least 50 should be there to actually get the total mRNA count of almost 2 lakhs, so that you can capture the entire mRNA or messenger RNA from a particular cell, right? And then since you are dealing it with mRNA, you have to use reverse transcription to convert that into cDNA, and then even in that step you may lose some information about the true number of copies that were present, because it is partly subjective to the reverse transcriptase and chance factor also, but still the overall representation of the different transcripts should be fine, right? And basically for a single cell transcriptome, if you want to do, so this was for the tissue if you see. So for the single cell, basically you need to follow the amplification step, like in case of chromium instrument also, which we will see later. One good way of separating or sorting the cells is micromanipulator, where you can do this manually. So basically this is more useful for the like bacterial cells which can pick under the microscope. So you can operate the micropipettes which can capture the single cells, right? So which it basically micromanipulator typically has a joystick and mechanism for reducing the range of movement, and then you can hold that micropipette to actually pick the or the microcapillary to pick the cells one by one. Okay. So microscope is needed because under which you can see. So 100-fold magnification microscope phase contrast is generally needed, and micromanipulator model for just one example is given, like but there could be more sophisticated micromanipulators also. Okay. So sterile glass capillaries with dimensions of micro size is needed to capture the microbes, okay, which is of about 5 micrometer, okay, and it should be at the angle of 45 degree to actually pick the cells one by one. So cells could be the dried bacteria cultures resuspended in a suitable sterile environment or nutrient, right, and which are actually put in the slide for the pickup. Like this is the dried bacterial culture in sterile solution. Okay, now okay, so this capillary also contain the sterile solution. So this can be taken up by the micromanipulator, okay, and it goes in and stays in this capillary here. Okay, so you can keep a few number of cells in this capillary, right, but you cannot keep too many because if you take any incorrect cell, then there could be an issue, and also it cannot hold too many cells. So once you have got like 15-20 cells, you can pick and put in a separate place, so that you can, because at least 100 or more cell you should pick up to go for the complete genome amplification for a single bacteria. Okay, so basically what it is doing is, under the microscope you are viewing the cells. So like this is one cell that you have found. So you will bring your microcapillary, okay, which is a very thin tube like structure, and attached to the micromanipulator, which is adjusting the pressure, and you have a joystick also, right, and you will be actually picking the cells from here, right, which will go in and stay inside the microcapillary. So same type of cells which you are seeing under the microscope, you can pick up and extract. So this is the micromanipulator, okay, and these are the arms, okay, 2 robotic arms and a joystick to actually capture the cells by actually looking under the microscope from here. So it is very good for basically isolating single microbes, and risk of contamination is also less because you are doing it manually, and although even if you have picked any incorrect cell, that may be removed later by even if it is sequenced, it can be removed later by computational method, but still like you can regulate or control this process of picking the right cells. Okay, and some microbes may be in very low numbers can also be examined, which may not get sufficient representation when you're dealing with the community together. Okay, and in case of aerobic it is easy, but in case of anaerobic where culturing is not possible or very tricky or required very tedious process, okay, this can directly go for the isolation and sequencing, right. So one example of micromanipulator I am just providing here. So like for example in the termite gut, if you see there are about 300 type of microbes which are there in the termite gut, and basically they are helping the termite to degrade the cellulose which it is eating all the time, right. So studying those bacteria inside the termite gut can tell you about the new enzymes which can degrade the cellulose better, new cellulases, right, or which can have like various translational applications. Okay, so basically a termite can provide almost 2 liters of hydrogen when it takes a single one A4 sheet of paper, okay, and which is done by, and that fermentation is done by the species that exists inside the termite. But it is not a simple system, and although it has 300 species, these two, RSG17 and CFPT1, are the most predominant inside the termite gut. So, but it is not so easy, because in the termite, if you isolate the gut, inside the gut there are protozoans, protists, and inside the protists there are basically these endosymbionts, okay, which are actually doing this job of metabolizing or fermenting that cellulose, okay. So basically in this case the bacteria were picked through the specialized procedure, that is the micromanipulator here. And then for when you have got about 100 cells, you can break open and do the whole genome amplification, isothermal, because you want to keep the temperature also constant. And once you have got sufficient DNA, you can go for the shotgun sequencing using NGS and reconstruct the genomes, okay. Like this could be this was the first genome that came from termite gut, and it belongs to an unculturable bacteria. It's an endosymbiont, so it has lost most of its function for independent survival. So the only way possible today to get access to its genetic information is by this method, like looking at isolating the single cell and sequencing it directly without actually culturing it. So it was a small genome of about 1.1 MB in size. A lot of genes that have become pseudo genes, right, of almost 121. Very small number of genes, about 800 genes, right, okay. So this was possible only because you could use it by micromanipulator, and later on the information that you got from the gene analysis and the genome analysis, you can construct a metabolic model and show that how this is actually what it is taking from the environment, okay, and what is giving out to the bacteria. Okay, another bacteria which was also abundant, that was also captured using a single cell genomics method by the micromanipulator, and that is analyzed, and in that basically the researchers could find that it contains after the genome construction and annotation, they found that this bacteria contains the genes to actually fix the environmental nitrogen, and also this bacteria is helping in recycling the nitrogenous compounds inside the bacteria. And the theory that emerged from this is that the termite do not eat any nitrogenous material, and basically it survives on wood, and these bacteria actually fix the environmental nitrogen and also recycle the nitrogen which is there in the termite gut for its survival. So these bacteria were crucial for their survival. So this was the NIF operon that was found in the termite gut bacteria, okay. And again, like kind of model how this bacteria is actually playing an important role in that microenvironment, okay. So this is an example of single cell genomics and its application, and then you can recapture how this bacteria is contributing to the termite and how it is adjusting or doing in that microenvironment. Okay, so far we have seen that how we can isolate single cells, we can do their gene expression, we can also analyze their genomes, right, which can help us to understand the role of cell in the context of the environment and in effect to some perturbations, or right, and also the way by which we can actually create the single cells and get their DNA. So now we can see that how this single cell genomics is important to understand the disease pathogenesis. For example, this is a tissue, right. So it may contain it's a organ which may contain several tissues, right. So you can do like this cell to cell communications, because it is important to understand that how in a complex system like a heart, the different tissues are helping each other and integrating, working together, right. So the cell to cell communication is very, very important, and that we can know only by single cell genomics. So far we have been generating data for the entire tissue where multiple cells were broken together, and all the mRNA genome get mixed up, and because the idea is that or the assumption is that the RNA profiles will also be same, although the genome will be same, right, but imprinting may be different, okay. So in single cell genomics we can understand the we are talking about each and every cell. So by knowing that, we can now interpret the cell to cell communications. Similarly, the spatial analysis, that how these cells in a given space are connected and related to each other or talking to each other, that is communication. And trajectory, like how they are dividing in a space, and so that processes can also be understood. Then basically to understand the epigenome, because each and every cell may have different methylation patterns, it will be good to create the methylome for each cell, and then see or ask the question whether the methylation patterns are different or same, or that may give rise to differential gene expression also. Okay. Similarly, like in case of immunological responses of cells, which is important, like in this case T cell receptors, where we can see that how these cells are responding to the immunological responses. Now this picture, they have tried to create a single cell type transcriptomics map of human tissues. Okay, so basically the figure says that annotating 51 cell types from 13 tissues using single cell transcription data. So data has been, like in this work, they have in the science advances recently, they have extracted the transcriptome from different cells from tissues, and from lung, heart, pancreas, liver, skin, testis, prostate, eye, kidney, small intestine, colon, rectum, placenta, and blood. So like that tissue that we know, right? And then from those they have extracted the cells, and using the transcriptional profiles from the different cells, they have clustered the cells based on their, you can see that in the figure B, UMAP plot showing the relationship of all cell types from all analyzed tissues. The color coding corresponds to 12 main cell type groups. So like this color coding that you see is from the main 12 cell types, and in the figure C you can see that cell type dendrogram showing the relationship between all 51 identified main single cell types based on genome-wide expression. Okay, so here you can see that how the we can cluster these cells, the 51 different type of single cell types, and understand the relationship between them. Okay, then this single cell gene expression heterogeneity in cancer can also be addressed by single cell RNA sequencing, like in this case this is a normal tissue, and whereas in cancer tissue basically may be very, very heterogeneous. So we can understand the heterogeneity better if we profile their gene expression patterns, and also the other information, right, which we can only do by single cell omics, right. Similarly, like patient stratification, responder versus non-responder, can also be done based on the single cell genomics, which can give you clues for various drug combinations that you can give, right, and also the to better understand the tumor microenvironment and cell to cell interactions, right. So basically single cell genomics, that is why is very, very important for the to understand the disease states. Okay, and ultimately, and this understanding will give rise to the development of personalized treatment, which is the ultimate aim and goal for the various therapeutics, which is mainly focused on the human health. So here what we are trying to understand is like first cell, and then how this cell is communicating or performing its role in the tissue environment, and which ultimately is helping or leading to the complexity that we see in the complete organism. Okay, so basically cells could be healthy or diseased, various heterogeneous, also various types of disease cells, right. So we can understand the cell identity, healthy and diseased, the state of the disease, the progression of the disease over a period of time, and which can help us to plan the personalized treatment for that given individual. So that is why this single cell omics will be very, very important. Okay. Now here what they have done is they have looked at the cellular heterogeneity in cancer by single cell transcriptome analysis. So basically if you see, the details are here. So they have they have done the clustering showing patient specific tumor groups in multiple samples. Okay, so basically these are the different samples obtained, and this color actually is by the sample, and compared to multi-patient clustering of normal and non-malignant cells, that is the orange dotted line, okay, which you see here, right. And from this basic data they are so basically this is the differences in the transcriptome data, and what they are inferring from this is the coffee number variations in the tumor cells and the intratumoral heterogeneity within a selected patient sample, and also the somatic mutations that separate a tumor cell cluster, right. You map and demonstrate the intratumoral heterogeneity in individual cells, the lower heat maps that we see in this picture, right, this one. So basically this can help us to perform complex analysis of understanding the heterogeneity in terms of the gene expression in like cancer versus the healthy cells. So now to perform the single cell genomics, a very good platform is the Chromium platform, okay, which basically deals with the processing of single cells, and also you can analyze the it can construct the library which can be actually used for later sequencing. So basically it is the based on gel in bead emulsion technology or GEM technology for the integrated analysis single cell at a massive scale. So you don't need to do the each and every cell separately, but it can take up thousands of cells and do the analysis. Okay, so thousands of single cell partitions with the barcode are available. So basically for each and every cell there will be separate barcoding of its content, including the DNA or RNA, right, and which can be linked to genome sequencing later. So there are two types of Chromium instrument till the date we were making this presentation, right, and one is partially manual, other is automated, and generally like these systems are not available in your individual lab, but you can always use it through a third party which is a service provider which deals with sequencing. Okay, so first of all you should have a cell or nuclei suspension, okay, and then it will, using the Chromium, it will construct the sequence ready libraries and followed by sequencing and the further downstream analysis. So we will see this process in detail. Basically it deals with the gel beads, cells or nuclei, okay. Basically beads, you can see these are the various beads, okay, in which basically these are the emulsions in which each emulsion will capture a single nuclei plus enzyme, right, for the downstream processing, okay. So within the Chromium instrument, barcoded gel beads are mixed with the cell or nuclei. So these are the barcoded gel beads, that is why they are shown in different colors, and the cell nuclei which are going through this single each. So basically the size wise they are ensuring that there is one cell nuclei and enzyme per bead, right. And then they can and partitioning oil to form tens and thousands of gems, and this way they can form thousands of them. So because you can see that there is this color and this is the bead, okay. In this it is captured nuclei, but here it is not captured, okay. So many cases may be like that. So it will but in this emulsion basically you can see that there is a bead, the nuclei and the enzyme, okay. So basically each gem acts as an individual reaction droplet. So this piece is acting as a reaction droplet where basically you can barcode the DNA fragments which can later be used for sequencing. So after barcoding, all fragments from the same cell or nuclei share a common 10x barcode. So in this there will be one barcode, in this there is one barcode. So basically we have separated the cells and attach the barcodes to each to the individual DNA extracted from each cell, right. So even if now we break it or do something, so basically you can see the 10 barcoded DNA fragments, you can pull now all these together because they are already color coded or barcoded differently, and then you can do pooling and sequencing of all the thousands of gen and their contents together. So generally it is followed by the Illumina sequencing to generate short reads which are later assembled. Okay, so ultimately basically you are dealing with individual nuclei which are forming single cell gems like this. Okay, and in each gem you can see that there is a gel bead, single cell, and basically reagents which will do all the processing inside this gel in bead emulsion, jet bead in emulsion. Okay, now in the same bead you can basically do the gene expression study also. So like which can provide a single cell transcriptome, okay, and to explore the cellular heterogeneity and to get more insights about the genic expression of each and every cell. Okay, so basically this is the functionalized sequences on the gel bead enable capture of 3' RNA alone in combination with feature barcoded antibodies. Okay, so basically here these are the sequences which are captured, okay, and which is attached with the barcodes, right. Now the next latest thing in this direction is the spatial transcriptomics, right, which can capture the like the context of transcriptional activity of the intact tissue. So now we are dealing with single cells separately, but can we really study all those cells, right, the way in which they are organized in the tissue, right. So if these are all the single cells, so can we study all these cells together in their real context without actually disturbing or isolating a single cell. So this is what spatial transcriptomics is, and which is very important and upcoming area. So for this, a tissue cryosection is attached to a spatial transcriptomic slide with barcoded primer. So the same context of single cell genomics. So okay, so you can overlap that. You know, first of all, the picture of this organization of cells, and on top of those cells you can basically overlap your transcriptomic slide that contains the barcoded primers. Okay, so for each and every tissue, each and every cell in that tissue, you can get the information. So basically when the tissue section attached to the slide, the reverse transcription of captured mRNA is initiated. So this picture can explain better. So this is a array of spatially barcoded mRNA capturing reverse transcription primers. So basically, okay, so this is the array. So in each one of these, there are individual spot contains millions of copies of an individual primer. Okay, so basically, and for each and every of these spot that you see, there is a mRNA capture where the mRNA will be there, the spatial ID, that is the barcode, which tells that okay, in the 3 dimension this is the spatial ID barcode and a RT primer. Okay, so now when this thing is the tissue is placed on top of it, basically due to the dimensions, size and matching of that, this can capture the contents from each and every cell, right. So this way basically the captured tissue mRNA will be taken up by the arrays of spatially barcoded primers, containing spatially barcode primers, and then you can see that in 3 dimension we can see now this is similar to the map of cells in the 3 dimensions, right, and we can check that how the mRNA expression is varying in that area. So okay, so for example, this has several applications, because here we are not only capturing a single cell but also in its real vicinity. So we can study the tissue homeostasis, like there could be some sites which maybe having some issues, or basically having differential gene expression, or have a propensity to become disease later. So these kind of insights can be get from this spatial transcriptomics. We can study the tissue development by overlapping the tissues with the array of those spots, right. The disease microenvironment in the real context, or tumor cell populations and their individual study. So basically this will help in a lot of things that we could not do by the conventional RNA sequencing or transcriptome sequencing or single cell genomics. Okay, So people have now made or trying to make the human spatial atlas, where basically they can see the spatial heterogeneity in diseases in the context of different cells, right? Now, for example, like there is a developmental spatial blueprint from the developmental stage of a single cell to a multicellular, and then how this is tissue expression varies in different tissues of that organism. And like similarly to identification of cell subpopulations, identify the different subtypes of the cell in 3 dimensions, physical localization of cell populations in tissue, like these spots that we saw in the previous picture, right? Overlap with the actual tissue, okay, where each and every spot is a separate barcode, and whatever they capture from that individual cell will be separately barcoded and later on analyzed, maybe in the similar way as the single cell genomics, right? And of course this may also have like to understand the intracellular communication also, it can help. So in summary, you can say that single cell omics is very important today, and it is a very important tool to understand the molecular basis of cellular mechanisms and refine our knowledge of the diverse cell states. It can reveal the heterogeneity of cells at the cellular level, because in a tissue different cells may be functioning slightly differently, which we can capture by single cell genomics. And the applications are definitely in diseases where we really observe that the changes in the individual cell functioning leads to some disease state, okay. And also this can single cell omics can help to understand the disease associated phenotypes or genes at to cell types to single cell level, right, to reveal the heterogeneity in the various cells. Similarly, single cell epigenomics can help to understand the differential methylation status of each cell, right? And for microbiome or bacterial or biofilm also this has applications, because in microbiome, like in a gut or a biofilm, different cells are doing different jobs, so you can capture the their differential genomes and the functioning also. So in this lecture, we have looked at the concept of single cell versus tissue in terms of genomic or transcriptomic sequencing, the single cell genomics, what it is and how it is performed, the different methods which can be used for doing single cell genomics, including the 10X Chromium link reads and Illumina sequencing, which can, so the Chromium instrument can prepare the library for each and every cell by using the GEM technology, and then that library can be later on used for the Illumina sequencing using short reads. And we also looked at the spatial transcriptomics and single cell gene expression, which is very important to understand the state of a tissue in real time. So this is good for today. Thank you.